Blood is considered to be fluid connective tissue because it contains cells, a liquid ground substance called plasma, and dissolved proteins. The functions of blood include transportation, where numerous elements and compounds throughout the body are transported via the blood. So, for example, erythrocytes and plasma carry oxygen from the lungs to the body cells and then transport the carbon dioxide produced in the cells back to the lungs to diffuse from the body. Blood plasma transports nutrients that have been absorbed from the GI tract, and plasma also transports hormones that are secreted by endocrine glands. Plasma also carries waste products from the cells to organs such as kidneys, where these can be later excreted. The blood also regulates many body functions such as body temperature, where plasma absorbs and distributes heat throughout the body. So if we need to be cooled off, then the blood vessels will dilate and then dissipate excess heat through the integument. Whereas when we need to conserve heat, the blood vessels constrict and the warm blood is shunted to deeper blood vessels in the body. Blood also helps to regulate pH levels in the body tissues, where we try to maintain a pH of 7.4, which is required for normal cellular functioning. If we have acidosis, this means that the blood pH falls below 7.0, and this can cause the central nervous system to be depressed, and so coma and death could occur. When we have alkalosis, which is a pH in blood that is above 7.8, this can create a hyperexcited state for the nervous system, and convulsions can occur due to that. The blood also maintains normal fluid levels in the cardiovascular system and to help prevent fluid loss. There is a constant exchange of fluid that takes place between the blood plasma and the interstitial fluid, such that if too much fluid is absorbed through the body, then we have high blood pressure that occurs, where if we have too much fluid that is escaped from the bloodstream and it enters the tissue, well then this causes low blood pressure, and so this causes swelling within the tissues. Blood contains molecules such as salts and proteins to help prevent this excess fluid loss from the plasma. The last function of blood is for protection, and so we have leukocytes that help to guard against infections by mounting an immune response if a pathogen or an antigen, which is a substance perceived as a foreign to the body, is found within the body. Antibodies then are molecules that are produced that can completely kill or remove the foreign object or antigen that are then transported in the plasma. Platelets and blood proteins can also protect against blood loss from forming blood clots. So within adults, about 4 to 6 liters is comprised of blood, which again is fluid connective tissue. And we can separate whole blood into its liquid and cellular components by centrifuge. And so that's what is outlined in the figure below. And so the steps involved having blood be sampled from a vein and then collected into a glass tube called a centrifuge tube. Then the glass tube is placed into the centrifuge, where it spins into a circular motion for several minutes. And then so that rotational movement helps to separate the blood into the liquid and cellular components based on its weight, and allowing the elements to be examined separately, which then we have about 45% of blood is consisted of formed elements, which make up our erythrocytes, or red blood cells, and plasma comprises about 55%, which is composed of many different proteins, water, and solutes, and then less than 1% of whole blood contains leukocytes and platelets, and that's what separates the plasma from the formed elements and is considered to be this buffy coat. Plasma is a complex mixture of water, proteins, and other solutes. And so the majority of plasma is consistent of water, which makes up about 92% of plasma, and it helps to act as a solvent for formed elements, solutes, and waste that are suspended in it. Then we have plasma proteins, which make up about 7% of plasma, where we have albumins, and these are the smallest and most abundant of the plasma proteins, where they make up about 58% of all plasma proteins. And what they do is help regulate water movement between the blood and interstitial fluid by osmosis. And then they also help to transport proteins that carry ions, hormones, and other lipids in the blood. Globulins are also proteins that are found within plasma, which make up about 37% of all plasma proteins. And what they do is that they help to act as defense cells to protect the body against pathogens that may cause diseases. Fibrinogen makes up about 4% of all plasma proteins and is responsible for blood clotting formation. And then our regulatory proteins are less than 1% of all plasma proteins that include the enzymes, proenzymes, and hormones that are being transported to other parts of the body. Then we have other solutes, so electrolytes, nutrients, respiratory gases, and waste make up less than about 1% of all plasma. So electrolytes include sodium, potassium, calcium, chloride, iron, bicarbonate, and hydrogen, and so they help to establish and maintain membrane potentials, help to maintain the pH, and to regulate osmosis. Nutrients such as amino acid, glucose, cholesterol, vitamins, and fatty acids are our energy source. Our respiratory gases, which are carbon dioxide and oxygen, and then our waste products, which are breakdown products of metabolism like lactic acid, creatinine, urea, bilirubin, and ammonia, are transported to the liver and kidneys where then they can be excreted. Serum is going to be our plasma, or part of plasma that has the clotting factors that are removed. And that is the only difference between plasma and serum. Formed elements are made of 3 components. The first is erythrocytes, which make up more than 99% of formed elements. And their primary function is to transport respiratory gases in the blood. Leukocytes make up less than 0.01% of formed elements and contribute to mounting an immune response and defending the body against pathogens. And then platelets, or thrombocytes, make up less than 1% of formed elements and help with blood clotting. The components of formed elements can be viewed using a blood smear, and so that's what the figure outlines below. And so what you do is you prick your finger, and a small amount of blood will be collected, and a blood drop then can be placed onto a glass slide, and then a second slide spreads the drop of the blood against the first slide, so smearing the thin surface of blood along the slide. And then the thin layer of blood then is stained to provide contrast for viewing the smear as the smear dries. So once it dries, then it's placed on a specimen to protect it, and then you can view it under a light microscope, so you can determine the number of erythrocytes and leukocytes as well as platelets that are found within your blood. Erythrocytes are commonly referred to as red blood cells. However, mature erythrocytes do not have a nucleus or any organelles. Erythrocytes transport oxygen and carbon dioxide to and from the tissues and the lungs. They have a biconcave disc structure, and this allows for respiratory gases to be loaded and unloaded rapidly and efficiently. Erythrocytes line up in a single file as they pass through small blood vessels. And so every erythrocyte is filled with approximately 280 million molecules of hemoglobin, which is a red pigmented protein. Hemoglobin transports oxygen and carbon dioxide and is responsible for the characteristic bright red color of arterial blood. When blood is maximally loaded with oxygen, it's termed oxygenated. When some oxygen is lost and carbon dioxide is gained during respiratory gas exchange, blood is then deoxygenated. And so deoxygenated blood has a deep red color that is perceived as blue when observed through the skin and the subcutaneous layer. On hemoglobin, there are non-protein groups, which are called hemes, and this is where we find an iron ion at its center, where this is where oxygen binds to iron for transportation of blood. So each hemoglobin molecule has 4 iron ions and is capable of binding 4 molecules of oxygen. The erythrocyte has a finite lifespan for about 180 days due to the lack of any organelles and to resynthesize proteins. And so old erythrocytes are phagocytized in the liver and spleen by cells called macrophages. The plasma membrane of an erythrocyte has numerous molecules called surface antigens that project from the plasma membrane surface. And this is where we have our ABO blood group. And there are 2 surface antigens that we have, called A and B. Blood with erythrocytes having surface antigen A is called type A blood. Blood with erythrocytes having surface antigen B is called type B blood. Blood with erythrocytes having surface antigens of A and B is called AB blood type. And blood with erythrocytes having neither antigen A or antigen B is called type O blood. And so with this, we have specific antibodies that travel into the blood plasma, and the antibody is a protein that is produced by a white blood cell, the B lymphocyte, and designed to recognize and immobilize a specific antigen it perceives as foreign to the body. So for instance, your blood plasma does not have an antibody that recognizes the surface antigens on your erythrocytes. Type A blood has anti-B antibodies in its blood plasma. Type B blood has anti-A antibodies in its B plasma. Type AB blood has neither anti-A nor anti-B antibodies in its blood plasma. And type O blood has both anti-A and anti-B antibodies in its blood plasma. And then another group, the Rh blood type, is determined by the presence or absence of the Rh surface antigen, often called either Rh factor or surface antigen D. When the Rh factor is present, the individual is said to be Rh positive, whereas if an individual who is Rh negative, then the surface antigen is lacking from the membrane of their erythrocytes. Antibodies to the Rh factor appear in the blood only when the Rh negative individual is exposed to Rh positive blood. So individuals who are Rh positive never exhibit Rh antibodies because they possess the Rh antigen on their erythrocyte. Only individuals who are Rh negative can exhibit Rh antibodies and can occur after exposure to the Rh antigen. So one clinically significant area of blood has to do with blood transfusion and something called agglutination. And so when a person is transfused with blood of an incompatible type, antibodies in the plasma bind to surface antigens of the transfused erythrocytes, and clumps of erythrocytes then bind together in a process called agglutination. So these clumped erythrocytes can block blood vessels and prevent the normal circulation of the blood. We have a universal blood type donor, which we consider to be type O, and then the universal recipient, which is type AB. So type O blood, again, has antibodies of anti-A and anti-B, whereas with type AB blood, we have neither anti-antibodies of A or B. Leukocytes, or white blood cells, help initiate an immune response and defend the body against different pathogens. And these are considered true cells because they contain a nucleus and cellular organelles. They also are 1.5 to 3 times larger than erythrocytes and do not contain hemoglobin. So we have two main groupings of leukocytes, granulocytes and agranulocytes. So granulocytes, which will be the first three leukocytes that we mentioned, have a granular appearance in their cytoplasm when viewed under a microscope. So the first granulocyte of a leukocyte is a neutrophil, and these are the most numerous, where they make up about 50 to 70% of all leukocytes. They have a light lilac-colored cytoplasm and a bilobed nucleus. Neutrophils usually remain in circulation for about 10 to 12 hours before they exit the blood vessels and enter the tissue spaces, where then they phagocytize infectious pathogens like bacteria. So how they attack bacteria is by secreting lysosomes, which helps to destroy the bacterial cell walls. And so whenever we're prone to bacterial infection, we have a significant increase in the number of neutrophils circulating within our bloodstream. Eosinophils have a reddish to a pink-orange granular color in their cytoplasm and only make up about 1 to 4% of all leukocytes. Their nucleus is also bilobed, and eosinophils increase in number when they encounter and react to phagocytized antigen-antibody complexes or allergens, which are antigens that initiate a hypersensitive or allergic reaction. If the body is infected by parasitic worms, eosinophils also chemically mediate and attack the worms. The last of the granulocytes is basophils, and these make up only 0.5 to 1% of all leukocytes. They too have a bilobed nucleus and a blue-violet granules found in the cytoplasm, which tend to cover up the nucleus under the microscope. These granules are histamine and heparin, which are released during anti-inflammatory or allergic reactions. When histamine is released from the granules, it causes an increase in the diameter of blood vessels, so causing vasodilation, and results in a decrease blood pressure, and with classic allergic symptoms like swollen nasal membranes, itchy runny noses, and watering eyes. The release of heparin from basophils inhibits blood clotting, which is termed anticoagulation. The next two types of leukocytes are agranulocytes because they do not have a grainy appearance in their cytoplasm when viewed under a microscope. So the first one is monocytes, and these make up 2 to 8% of all leukocytes. Their nucleus has a kidney shape or a C shape to it, and after 3 days in circulation, monocytes exit blood vessels and take up residence in tissues, where then they change into large phagocytic cells that are termed macrophages. Macrophages then phagocytize bacteria, cell fragments, dead cells, and different debris caused by other cells within the body. Lymphocytes reside in lymphatic organs and structures and make up 20 to 40% of the number of leukocytes that we have. They have a dark staining nucleus that basically takes up almost the whole cell. When activated, lymphocytes grow in size and have proportionally more cytoplasm. There are three main categories of lymphocytes. We have T lymphocytes, which manage and direct an immune response, and some even attack foreign cells and virus-infected cells. B lymphocytes, which are stimulated to become plasma cells and to produce antibodies, and natural killer cells, which attack abnormal and infected tissue cells. Platelets are irregular, membrane-enclosed cellular fragments that have never had a nucleus. Sometimes they're referred to as thrombocytes, but that's inaccurate because they've never been cells. Platelets are continuously produced in the red bone marrow by cells called megakaryocytes. Megakaryocytes are large in size, and they have a dense multilobed nucleus. They extend these long processes throughout the blood plasma wall. So what this does is it helps to form and initiate blood clotting. where, for instance, if somebody had undergone severe trauma to a blood vessel, this causes the blood to coagulate or clot due to a web of fibrin that helps trap erythrocytes and platelets to halt blood flow. Due to the relatively short lifespan of formed elements, we have a continual production of new elements called hemopoiesis. And so this is where we have blood cells that are continuously being replaced and formed in the red bone marrow. And so since the stem cells are the ones that start the initiation process, these can then develop into erythrocytes, thrombocytes, or leukocytes. And so we have about 8 million red blood cells that are produced in the bone marrow every single second. Some clinical conditions that occur with abnormal blood cell formation is leukemia. And so leukemia is a cancer of the leukocyte forming cells, where there are many different forms of leukemia, but all of them have an abnormal development and proliferation of leukocytes in both the bone marrow and in circulating blood. And so they're classified on their duration of either acute or chronic.